So welcome everybody to Talking Public Health. Today, we're going to hear from Dr. Christina Stallings, who is the Theodore and Bertha Bryan Professor in Environmental Medicine in the Department of Molecular Microbiology. She's also the founder and scientific director of the Mycobacteria Focus Program for Research and Innovation in Science and Medicine, and the co-founder of the Philip and Seema Needleman Center for Autophagy, Therapeutics and Research at School of Medicine. That's quite a lot. I'm starting to feel really bad. She received her PhD with distinction from Columbia University College of Physicians and Surgeons and transitioned her graduate work to studying chronic pathogens, Mycobacterium tuberculosis for her postdoctoral research at Sloan Kettering. And she began at WashU in 2010. And her lab seeks to dissect molecular mechanisms involved in tuberculosis from the perspective of the host and the pathogen. She's been recognized for her accomplishments in these areas by being elected to the American Academy of Microbiology. She's been awarded a Burroughs Wellcome Fund Investigator in the Pathogenesis of Infectious Disease, and is the Arnold and Mabel Beckman Foundation, has an Arnold and Mabel Beckman Foundation Young Investigator Award and the American Lung Association Young Investigator Award as a TB scholar. She is dedicated to translating basic science findings to the development of new therapeutics and currently holds 5 patents for the development of new antibiotics to treat TB. She's committed to mentoring and promoting the success of the next generation of scientists and works hard to increase accessibility and diversity in STEM careers by serving as a mentor for the Compass Training and Mentoring Program for Young Investigators associated with the NIH Mosaic program. So with that, very impressive indeed. We look forward to hearing from Dr. Stallings.
Thank you for that great intro. So I think I'm probably a pretty atypical speaker here, because I actually don't do public health or global health. But really the reason I'm here is I want to get you excited about mycobacteria from a public health and global health perspective. And I'm really interested just in general in making more connections in that direction and in that aspect. So this is my goal for today. And a lot of what I'm going to talk about is tuberculosis, which is a major global health crisis. It kills more people than any other infection in the world, and we'll go through that. But I actually first want to take a step back and talk about mycobacteria in general, because Mycobacterium tuberculosis isn't the only relevant mycobacteria for human health. And so this is what mycobacteria look like. So they're pretty boring. They're just these rod-shaped bacteria. They don't have any of the cool stuff that people look for on bacteria, like flagella or pili or toxins or any of this stuff. This is it. And this is actually the giant microbe version of Mycobacterium tuberculosis. And it's like the most boring giant microbe that they sell, right? It's just a stuffed tube. But despite being really boring looking, I'm going to tell you that they're not so boring in general, though. And I just want to point out that there are close to 200 known species of mycobacteria. And so we're going to talk about 3 today, but this really is like a tremendously large class of, or genus of bacteria. And what makes mycobacteria special is their outer cell envelope. So mycobacteria are coated in this cell envelope, which has these really, really thick, long chain fatty acids. And so it's really this waxy barrier that this bacteria has around it. And the reason that this is relevant for health is that this barrier, as you can imagine, makes the bacteria really resistant to a lot of things. So these bacteria are naturally resistant to a lot of antibiotics, as well as other stresses like osmotic stress or reactive oxygen species. This barrier for pathogens can also modulate immune responses, and it helps the bacteria form biofilms, so layers on surfaces, and makes the bacteria impermeable to dyes and stains. And so this is just showing some of the properties of mycobacteria that can occur because they have this waxy coat. So cording, which is the picture on my title slide. So the bacteria will naturally come together in these serpentine-like cords. This is Mycobacterium tuberculosis. So it looks like super cool and beautiful, but that's also a very deadly picture right there. And they also have this really distinct colony morphology, which is actually used even still today to diagnose them as sometimes. And so normally when we think about diagnosing bacterial infections, we think about gram-positive or gram-negative bacteria, which are stained like you see here. But the thing is, if you have a sample from a person and you're trying to gram stain it, if it has mycobacteria in it, they're not going to stain because they have this waxy coat, right? And so in order for us to use diagnostic, at least staining microscopy tools to see whether or not someone has mycobacteria in a patient sample, we have this special type of dye that's called acid-fast staining. And so still now, the point we're at, where we've had mycobacteria with humans for a long time, the gold standard of actually diagnosing someone with mycobacteria is doing a smear on a slide and staining it with acid-fast staining to see if you see acid-fast positive bacteria. And we'll talk a little bit more about how like our gaps in diagnostic tools are also a really big barrier that we have in controlling these infections. Okay, so we're gonna focus on mycobacteria that are relevant to human health, but the vast majority of mycobacteria are not pathogens, and they don't cause diseases in humans. So they're just everywhere. They're in the soil, there's probably some on your bodies right now, they're all over the place. And so that's most mycobacteria. But it turns out some have evolved to cause some notable diseases. So the first one that's notable is Mycobacterium leprae causes the disease leprosy. And so I'm not going to show any pictures of leprosy, but I will show you the not so cuddly animal that carries leprosy around our country, which is the armadillo. And so this is not an irrelevant pathogen for our local area because with global warming, armadillos are moving north. And so if you drive just on 55 South a little, you might see some armadillo roadkill. And those armadillos probably have Mycobacterium leprae. So it's really endemic in this population. Don't play with armadillos because you might get leprosy. And so we do get some cases in the hospital here. It is very treatable though. But it still is a notable infection. The next one that I'll spend most of the time with is Mycobacterium tuberculosis. So this can be caused by a complex of bacteria shown here, but most commonly is caused by the eponymous Mycobacterium tuberculosis. And I'll talk a lot about this later in the slides. But the one that I actually want to start with is these non-tuberculosis mycobacteria. So this is a relatively large group of bacteria that can cause disease in humans, but are not tuberculosis or Mycobacterium leprae. And these generally are opportunistic pathogens. So normally they're just present in the soil, they're not bothering anyone, but if someone in one way or another is susceptible to them, they could cause disease. And they tend to be able to cause very, very difficult to treat diseases. And the reason I'm going to start with this is that we're going to talk about how tuberculosis, definitely there are cases in the US, but it's really considered more of a global health problem because the burden of TB is predominantly in other countries. Whereas with non-tuberculosis mycobacteria, this is a problem right here in our backyard. And so this is something that the hospital deals with constantly. And the predominant infection that's caused by non-tuberculosis mycobacteria are pulmonary infections. And so in the United States, our case prevalence is about 11.7 cases per 100,000 people. There are some areas of the world that have really pretty high prevalence rates, including areas in Asia. And usually when you have an infection with these non-tuberculosis mycobacteria, it's because you have some other underlying condition. And because it's generally a pulmonary infection, these tend to be diseases that are associated with pulmonary dysfunction. And so, for example, cystic fibrosis patients are very susceptible to non-tuberculosis mycobacterial infections. And then also COPD patients, as you get older, there are also associations. So many of you will at one point probably know someone who's older who has a non-tuberculosis mycobacteria pulmonary infection. So it's not uncommon. And so we're seeing these in our hospital a lot in our cystic fibrosis patients, our bronchiectasis patients, as well as in our transplant patients, so people that are immunocompromised. The issue with these infections is that there's a particular non-tuberculosis mycobacteria, which is Mycobacterium abscessus. And this is really an emerging public and global health threat that we have, because this is a otherwise environmental mycobacteria. It's in people's households, it's around, but it causes a very, very difficult to treat infection in patients that are vulnerable. And so if someone with, for instance, cystic fibrosis gets Mycobacterium abscessus infection, it's almost impossible to treat, which means that if they were going to get a lung transplant, they can't. So it takes them off these lists. And even with the new cystic fibrosis channel modulators, those patients aren't reversing the lung damage, so they're still susceptible to non-tuberculosis mycobacteria. And just to give you an idea of what happens if you have one of these infections in terms of treatment, the current treatment guidelines for someone with Mycobacterium abscessus is first an intensive phase of antibiotics, where you have an oral microlid in conjunction with 2 intravenous antibiotics for the duration dependent on the response to therapy, which often can be like over a year. And then you have a continuation period with multiple antibiotics that lasts for another year. And this is really a best case scenario. And then you have a 30% chance, dependent on your other conditions, of actually getting rid of these infections. And so, as you can imagine, this long-term antibiotic usage also has a lot of side effects related to it. There's a lot of patient adherence problems, and it really can be quite horrible for the patient burden. And we're really just at the beginning stages of understanding these infections, and we really know very little. So first of all, we don't know how people exactly are acquiring the infections. And so it's definitely been identified that these bacteria can be cultured out of like showers and bathtubs and sinks, so kind of these water sources in households. But these don't necessarily match with what we're seeing in patients as well. There's really not many case studies on people actually spreading it from person to person, but those are popping up a little bit more now, so that is possible. But we really even just have this gap in like, how do we control an infection where we don't know how people are getting it, right? And we don't know how they're exposing to it. So then it makes it really hard, as you can imagine, to protect them from it. There's also issues in terms of like testing for drug resistance and other problems as well with these infections. And so I only bring this up because we have a lot of interest, in general in the mycobacterial field of really starting to delve more into this issue of Mycobacterium abscessus and other non-tuberculosis mycobacteria because it is this emerging health threat. And we know so little right now. Okay, so now I'm going to switch to what's going to be the star of the rest of our show, which is Mycobacterium tuberculosis. And this really tends to take the limelight for mycobacteria because of its incredible burden on human health globally. And so tuberculosis has been with humans forever. It's an ancient disease. So if they take old mummified remains and sequence them now, because we sequence everything, right? We're like, oh, what's the sequence of this mummy? That turns probably that mummy had tuberculosis. And because tuberculosis DNA is so stable, when they sequence these remains, they come out 99% tuberculosis, 1% human. And so it really they can detect it from long, long, long ago, and the DNA can stick around for reasons we don't know. But you can see like old drawings, there's of course old paintings, these are old posters trying to tell people to be careful about tuberculosis. So it's been here forever. Oh yeah, and I'll say like there's a report that they think Mycobacterium tuberculosis actually evolved from environmental mycobacteria when our ancient human ancestors started using controlled fire. And the fire and the movement of the air moved the environmental mycobacteria up into our lungs. And like slowly then that started evolving to Mycobacterium tuberculosis. All right, so despite the fact that it's been here forever, we've also been co-evolving with Mycobacterium tuberculosis. It still is the most deadly infection in the world. And so it's about 1 person every 25 seconds in the world dies of tuberculosis. And so it's about 10.8 million cases a year, about 1.3 million deaths a year. And this is the global counts. We do have cases here in the United States as well, which are unfortunately on the rise. So there's about 11,000 cases of TB a year in the United States and about 600 deaths. And we'll talk about why these cases in the United States are lower than other places. But another really significant aspect of Mycobacterium tuberculosis is that it is the leading cause of death in HIV/AIDS patients as well. And so even HIV patients on antiretroviral therapy are still more susceptible to Mycobacterium tuberculosis infection. And so there's still something about their immune system that still increases the susceptibility. So as I mentioned before, most of this burden of tuberculosis is globally, but we do have cases in the United States. And so I'm just gonna focus a little bit on the US in terms of what's going on with TB there. And so first looking in the past, because I think we can learn a lot there. So this is looking at the incidence of tuberculosis up to 2015. And so you can see that there's this really prominent spike that occurred here in the early 90s. And so when we look at why that spike occurred, as you can probably speculate, it's because of the HIV/AIDS epidemic. And so what happened is that as a world, we thought we had TB under control. We had antibiotics, there were less cases. I mean, that was a tremendous advance to get antibiotic therapy. And then all these people developed AIDS and started reactivating TB. And it turned out it was in them the whole time and we didn't realize it. And we had cut like so many programs at that point because we thought tuberculosis was no longer a problem. And it led to this large spike globally and in the United States of tuberculosis cases. And so I think the other thing though, is on the other part of this slide is this decline. And this is what I really want to highlight, particularly in terms of this audience, in terms of what actually caused this decline. And the biggest thing that happened, there was antiretroviral therapy, but you still had all of these people with active TB spreading disease. The biggest thing that happened was public health. And so this was really the start of still some of our fundamental public health programs and global health programs that we have to control TB incidence. And the predominant, if not only reason that we had that drop at that time was because of public health initiatives. There were no other major medical advances in that time beyond antiretroviral therapy that could have led to this. And so then and even now, things that we do is that we focus on promptly identifying people with TB, initiating the appropriate treatment immediately, and then ensuring completion of the treatment. And this is critical. And so this really started this program called DOTS, which is Directed Observed Therapy Short Course, which has 5 elements. And in a country like the United States, this works really well. So you basically identify the person, get them on treatment, watch them take their treatment until they go to completion. And this is why in this public health focus on tuberculosis is why we've been able to keep the numbers low in the United States. Otherwise, we would be in a similar problem as anywhere else. And so if we look now where we're at today, and so this is going a little bit forward in that timeline. So you can see there was a dip in 2020, but that's assumed to be because people weren't going to the doctor to get diagnosed because of the COVID epidemic. But what you can see now is that we're having this increase in TB cases. And we're now at levels that are even higher than pre-COVID pandemic levels in the United States. And this is not just in the United States, this is globally. And just in the last couple of years, we had an outbreak in Kansas City, which some of you may have heard about, where there were 65 cases in one community of tuberculosis. And so it's originally, a lot of the thinking was that we were having this increase because people weren't getting diagnosed and getting treated during the height of the epidemic. And so the longer you go untreated with tuberculosis, the longer you spread the bacteria and the more people that get infected, right? And so it could be lag from that, but because the numbers keep rising to those pre-pandemic levels, there's some concerns that there's other things going on. And so I'm going to talk a little bit now then about how tuberculosis infection occurs. And so Mycobacterium tuberculosis, similar to the other mycobacteria, is generally a pulmonary infection. But what's different about Mycobacterium tuberculosis is that it is spread primarily, almost predominantly, by person-to-person transmission through aerosol transmission. And so if someone has active TB disease, so they're sick, they're coughing up the bacteria, and then someone else could be nearby that's unfortunate enough to breathe it in. Once they do that, the bacteria travel to the lower airways, so it's called the alveolar space, And we have these macrophages, these innate immune cells in our alveoli that are our sentinels of our lung. And anything that gets into our lungs, they eat. So they get rid of pollution, they get rid of bacteria, viruses, anything, cigarette, tar. Those alveolar macrophages are just sitting there eating whatever we breathe in. And so that includes the Mycobacterium tuberculosis. So those alveolar macrophages will eat or phagocytose that bacteria. But those cells, they can't control the infection. So mycobacteria is very good at growing in these cells. And so what happens is those alveolar macrophages will travel to the lung tissue and they'll alert the rest of the immune system that they cannot control this infection and they need help. And so that draws many other types of immune cells to the site of the infection, one of which I'll talk about at the very end is this adorable little immune cell, the neutrophil. But there are many other ones that come, like other macrophages, monocytes, dendritic cells. And so that originally infected alveolar macrophage dies from the infection and ends up spreading the bacteria to all these other cells. And then mycobacteria just grows in all of those other cells. And then what has to happen is dendritic cells will traffic to the lymph node, present antigen to these cells called CD4 T cells. CD4 T cells will then come back to the lung and activate those infected cells to control the bacteria. And so the reason that, one of the primary reasons that people that develop AIDS are so susceptible to TB is because their CD4 T cell counts drop and they no longer have this CD4 T cell mediated control of Mycobacterium tuberculosis infection, because this is absolutely essential. And what happens is once these CD4 T cells are in the lung, they cause the immune system to surround the bacteria in a bit of like a jail cell. That's this characteristic histological structure of Mycobacterium tuberculosis infection called a granuloma. And so in the vast majority of people that are infected with TB, in 90% of people that have good antibody responses, good T cell responses, they control this Mycobacterium tuberculosis infection very well. So it either stays within this granuloma, within this jail cell, or the bacteria gets completely cleared. The issue is that in about 10% of people that seem like they should be able to control, they don't. And this granuloma does not contain the bacteria. The bacteria are able to break out and then cause the symptoms that we associate with TB disease, like coughing up blood, fever, malaise, all the things you read about in old novels or hear about in old operas. And so all these symptoms, and it's these symptoms that are active TB cases, which contributed to about 10 million people falling ill and about 1.6 million TB-related deaths. And so when someone has active TB disease, they're coughing, they're sick, they go to the doctor, right? And then they start taking antibiotics. So very similar to the cases with non-tuberculosis mycobacteria is that Mycobacterium tuberculosis is also really recalcitrant to treatment, which means the courses of therapy are incredibly long. And so if you have drug-sensitive Mycobacterium tuberculosis infection, which is the best kind you can get, because you definitely don't want drug-resistant. So you have drug-sensitive Mycobacterium tuberculosis infection, active TB disease, you're going on about 6 months of multiple antibiotics. And so again, there's this intensive phase where you'll take 4 antibiotics at a time, and then a continuation phase where you'll have 2 to 3 antibiotics depending on your case. And these antibiotics are not fun to be on. This is not amoxicillin. These are things that are liver toxic. Rifampicin, one of our frontline antibiotics, turns like everything yellow, your eyes yellow, everything that comes out of your body yellow. And so it's really hard for the patients to be on these antibiotics as well, even though they want to get better. They're also counterindicative, rifampicin in particular, which is one of our frontline antibiotics, to antiretroviral, to protease inhibitors as well. And so here we have one of our predominantly susceptible populations, our HIV patients on ART, and now you're trying to treat them with TB and you can't use one of our frontline antibiotics because you don't want to compromise their ART therapy. And so because of this, really big burden on our patients, there of course is some patient adherence issues just because they can't actually take the drugs. They're just not, they're getting sicker on the drugs and they're getting better. And so because of this, we're getting this rise in drug-resistant tuberculosis. So antibiotic resistance is a big problem in general. I'm sure you guys hear a lot about it. TB is the #1 killer of antibiotic-resistant bacteria. And so this is a huge problem. And so another one of our frontline antibiotics, isoniazid, it's estimated that 1.3 million TB cases are resistant to this frontline antibiotic a year. And then this is associated with multi-drug resistance, which is also resistant to rifampicin. And there's about 500,000 of multi-drug resistant TB cases a year. So if you have drug-resistant TB, you can't take rifampicin and isoniazid. You have to take the second-line antibiotics. And these are often injectable, and you have to take them for multiple years. And you have about a 50% chance that you'll get cured. And so it's this infograph here, I feel like really hits to me what this means. So it's estimated that a patient that has drug-resistant TB, the number of pills that they have to take would stack to the height of the Golden Gate Bridge. This is a huge number of pills, a very tall bridge. It's so a huge burden on our patients, but also a really big burden on the healthcare system as well, as you can imagine. And again, a lot of these initially will be injectables as well. So you're talking about another burden on the healthcare system. And if you ever want to hear more about this, there are lots of TB testimonials that you can listen to, including through the CDC, the WHO, as well as other things that you can hear more. There's also a recent book out by John Green called Everything is Tuberculosis. I highly recommend it. John Green is not a scientist. So it's like he's a very famous young adult author, so a very good, author, and fell in love with tuberculosis in like a bad, wants to kill it way. And and wrote this really phenomenal book that I think is just very accessible to a lot of people. And that also talks a lot about that patient experience as well with patients that he met. Okay, so a lot of things. So the antibiotics, we have them, but they're really hard for the patients to take. And of course, like what we all want is a vaccine. We all want to just protect everyone, right? So it turns out there's been a TB vaccine around for a very, very long time, which is called BCG. And this vaccine was first developed in 1921, and we have not progressed past this technology yet, 100 years later. And this vaccine is an attenuated strain of another Mycobacterium bovis, and the virulent Mycobacterium bovis causes tuberculosis as well. It generally causes disseminated infection. It's carried in like cattle. This is why you should pasteurize your milk. It's not the only reason you should pasteurize your milk, but it is a reason you should pasteurize your milk before you drink it. But because it can cause very deadly Mycobacterium bovis. But this is an attenuated strain that was developed to be a vaccine. And what we see is that this vaccine is very good at preventing children from getting extrapulmonary disease, so TB infection outside of their lungs. Because what happens is children are very susceptible to Mycobacterium tuberculosis infection, but not only are they susceptible to infection, they tend to actually develop disease outside the lungs. So their lungs don't contain the infection, and it spreads to either their central nervous system and causes meningitis or to other sites in the body. And so if you vaccinate children with BCG, you're able to largely protect them from these extrapulmonary cases. And this is really, really important. So areas of the world where TB is endemic, they use the BCG vaccine to protect children. And this is a critical thing to do in terms of protecting against pediatric tuberculosis. The issue is that as people get older, those rates of extrapulmonary TB are very rare in adults, unless you are immunocompromised, like you have HIV, AIDS, or other issues. But generally, adults contain the bacteria in their lungs, and the vaccine has absolutely no efficacy against developing pulmonary TB as an adult. So you can get boosted as many times as you want, and it will not protect you against pulmonary TB as an adult. So we still, to this day, do not have a protective vaccine against TB. And this is just showing some of the data for that if you vaccinate, this is monkeys, but we see the same thing in humans, with BCG, you can completely protect at non-pulmonary sites like the spleen or the liver, but no effect in the lung. Okay, so because of all of this, the WHO developed this end tuberculosis strategy called WHO NTB. And so this was adopted in 2014. By the 67th World Health Assembly meeting, and the ultimate goal was by 2035 to have fewer than 10 new TB cases per 100,000 people, annually, which would amount to a 90% reduction in TB incidence and a 95% reduction in TB deaths. And the goal was to develop new methods of diagnosis, treatment, and also trying to find some new vaccine candidates and addressing many of the social determinants of TB. Because the other thing that we face in this country and others is that TB, similar to HIV, is a very stigmatized disease. So it's considered a disease of the poor, or of the people that have other things like HIV. And so we still wrestle with getting people to go to the doctor, even in this country, because of the stigma behind tuberculosis. And so really the idea was to tackle all of these things. And so just to show you where we're at here, so this is our goal line in blue. In order to reach this WHO NTB goal by 2035, and this is the trajectory that we were on. And now these rates are actually increasing. So instead of us going in this downward direction, where if we're lucky, we're plateauing, but at worst case scenario, we're actually increasing. Okay, so what do we need? What do we need to combat TB? We need everything. And this is why I want more public health people and global health people to be thinking about this. We need better epidemiology and better reporting. We need better diagnostic tests that are point of care tests. So they need to be something that's cheap, easy, that can be done anywhere in the world, right? Not just here at WashU. We could do any diagnostic test here, but we need something that we can do globally. We need a vaccine that protects against pulmonary tuberculosis. We need new treatment strategies that allow for shorter and safer multi-drug regimens against both drug-sensitive and drug-resistant TB. And we need more emphasis on prevention as well. And so with all of this in mind and just general interest in mycobacteria, I'm not the only person here at WashU who's noticed that mycobacteria is a problem and has gotten interested in it. So obviously the people in the clinic and the School of Medicine are very interested in especially non-tuberculosis mycobacteria, but there are a lot of engineers that are looking at like aerosol spread and looking at non-tuberculosis mycobacteria, people working on adjuvants, people developing new drugs against TB. And so what we are now launching here at WashU to try to start doing our part to address this global mycobacterial problem is this new initiative called MycoPrism. And Deborah kind of mentioned this in my bio. And so we're going to announce this very soon. So this is the website that we are announcing, and the School of Public Health is already on there as a partner. And the idea here is to really bring together people at WashU across all different areas to be thinking about and working on tackling these issues that surround mycobacterial disease in humans. And so I'm here because a key aspect of this, as I really highlighted, is that the way we have been able to get any handle on TB in the absence of having a protective vaccine is through public health and global health. And so having more people in that arena that are interested in this and working on this is only going to be good for us. And so we're really trying to bridge more opportunities to be able to interact more on that level. And what you'll also, everyone here will get this announcement because I'm going to send it to the someone who distributes things to you. But as part of announcing the website and this initiative, we're also going to have our launch symposium this spring, which will be in April. And so here, again, we want to highlight all these different ways and perspectives that we need to be thinking about mycobacteria in order to tackle these problems. And so I encourage you to pay attention to the email, possibly register. It's going to be at the Neuroscience building, so like halfway between you guys and us at the School of Medicine, or here in the School of Medicine. So it should be pretty easy to attend. Okay, so then I'm going to take a little bit at the end, because I can't help it, to talk a little bit about the work I do, which I'm going to show you some pretty pictures. And so we don't have to get into too much of the nitty gritty of the molecular details, but I just want to say a little bit that my lab is doing in order to try to fight these infections. And so if we go back to what do we need to combat TB? So as mentioned in the bio, I have a basic science laboratory. We molecularly dissect the pathogenesis of mycobacterial diseases, but we do have this commitment to trying to translate our work. And so we want to find new therapies and new treatments to treat tuberculosis. And so one of the things that we're interested in is whether or not we can modulate the immune response to better control TB. And the reason that we're interested in this is actually we know it can happen, because in 90% of the people that get sick, they do control the infection. So that means 90% of the time, just like TB has been evolving with us, we've evolved to contain it. So this is possible. All we need to do is figure out how these individuals are controlling, what's going wrong with the individuals that are developing active TB disease, and then manipulate their immune system so that they have this protective response. So basically skew them to be able to protect themselves. And the other reason that we really like this approach is that when you treat TB with antibiotics, you're going to select for antibiotic resistance, but this is going to be able to target the bacteria regardless of whether or not it has resistant genes or not, right? Because this is just going to be giving your immune system what it needs to be able to fight and survive. And so we looked into the clinical studies to see, like, what do we know that's different in the cases where people are able to protect versus people that are not able to protect. And one of the big things that comes out is the presence of this one innate immune cell type called the neutrophil. So this is the one that I referred to earlier, which was adorable looking, and you're going to see more of it and you'll see how gorgeous it is. But it has this association with active TB disease. And so if we take patients and look in their airways, so we either have sputum samples, bronchoalveolar lavage samples, or we look in necrotic lung tissue of these patients, what we see is that the predominant cell there is the neutrophil. So these are all patients with active TB disease. These are all patients that have not been able to control the infection. And then they have this association with these neutrophils accumulating in their lungs. And it's not just that the neutrophil is the predominant cell that's present, it's also the predominantly infected cell. And so that's where the bacteria is. It's in the neutrophil. And so this has been known for quite a while because we've been collecting things out of the airways of TB patients as long as we could and looking at there and trying to figure things out. But no one has really understood what that means, because the neutrophil, this is a very important immune cell in your bodies. It is the most common cell that white blood cell that you have. So it is all through your blood. You never want to be neutropenic because it makes you very, very susceptible to infections. So neutrophils are your professional killing cells. This is what they do. Anything that tries to invade you, they kill. And they do this by doing a lot of different things. So they will phagocytose or eat whatever invading pathogen is there, and then they'll attack the pathogen with bleach, basically, within these granules inside of the cell. They'll also release all these grenades and bombs called degranulation, full of antimicrobial things. And they'll release DNA that is just a spider web of DNA that's studded with antimicrobial effectors. And so it's like a web of antibiotics, basically. And so neutrophils do all of this, and yet they're there when these patients are not controlling the infection. And so we were curious whether or not they could control TB. Were they actually helping, or are they just there? And so in order to look at that, we collected neutrophils either from the blood of people or the bone marrow from mice. And then we just infected them with Mycobacterium tuberculosis, and we just watched to see if they could kill the bacteria. And so when we monitor the growth of the bacteria in neutrophils, we see that the bacteria grows great. So the neutrophil is not able to control the bacteria. It's not able to kill Mycobacterium tuberculosis. And in fact, Mycobacterium tuberculosis seems to grow in the presence of these neutrophils better than it grows in even in culture that we make in the lab. So we can't even recreate how wonderful of an environment this is for the bacteria. And so this is very counterintuitive, because this is the cell is what it's supposed to do, is to kill bacteria. So we wanted to understand more how the neutrophils were actually promoting the growth of Mycobacterium tuberculosis instead of killing it. And one of the things that we discovered was that in response to Mycobacterium tuberculosis infection, one of the things that neutrophils do is they release these neutrophil extracellular traps. So these are these spider webs that I mentioned of DNA. And what will happen is they'll eat Mycobacterium tuberculosis, they'll spew out these spider webs that are supposed to kill the bacteria, but instead what we see is the bacteria thrives on them. And so we wanted to study this more and see if this was actually happening in people when the bacteria was not being controlled. And so we're actually able to monitor this process with an antibody that detects the first step of this net release, which is called histone citrullination. And what that does is it changes the charge of the histones that your DNA is wrapped around. The DNA unravels so it can be released. So we can detect citrulinated histone 3 or H3-CYP. So then we looked in the lesions. So this is showing non-human primates that are infected with Mycobacterium tuberculosis, but similar things have been seen in human resected lung tissue from patients that are not responding to antibiotic therapy. And so this is what that TB granuloma, that lesion looks like. And you could have different types. You can have non-necrotic, which is associated with controlling the bacteria, or you can have this necrotic lesion, which is where the tissue, the lung actually just starts to melt. It just starts to degrade. There's no more cells there. The bacteria are just hanging out in this acellular compartment. And it's this necrotic lesion that then eventually breaks into the airways and can spread disease. And so it's associated with poor control of TB. And what we found is when we look specifically for neutrophils, which is this S100A9, or specifically for that NET marker, this H3-CYP, that we saw higher amounts of neutrophils and NETs in these lesions that were associated with poor control of TB disease. So again, these are the ones that you have lots of bacteria and it's going to spread the bacteria. And so we wanted to identify what was going on in this process of net release during TB infection so we could determine if we could try to modulate it to be able to treat tuberculosis. And so in order to do this, we went in the cell culture dish, so in vitro, to see if we could recapitulate it, and we can. So if we take neutrophils, again, we can do this out of human blood or mouse bone marrow, infect them with TB, and then we can see that they do citrullinate their histones and release these nets coming out of them. So these cells are releasing these strings of DNA. And what we find is that these strings of DNA look very different than if you induce them in other ways. And so you can chemically induce neutrophils to release these nets of antimicrobial webs of DNA. And that's shown here with PMA and ionomycin. And you get these thick explosion bundles coming out of the neutrophil. But during TB infection, we have this release of the DNA, but they're very, very, very thin strands. And you can actually see this. This is a scanning electron micrograph of a neutrophil. So this is one of your blood cells. And what we find is when we infect them with TB, you can see they're releasing these thin strands of DNA. And so the rest of the cell looks fine. It's just somehow able to take some of its DNA and just kick it out in this thin strand, but otherwise maintain its physiological appearance. And along with that, the way that, because it's able to do this, it actually maintains its viability. So this zombie dye stains dead cells. And what we find is when we infect with TB, we have all these neutrophils that are releasing their webs of DNA, but they're not dying. And this is different than what we see if we chemically induce the release of these webs. And so what we found is that the way that this is working, so this is a transmission electron microscopy of a neutrophil. And so this is a normal neutrophil straight out of the blood or the bone marrow of a mouse. And this is what it looks like. And then when we infect with TB, we get the buildup of these vesicles here. And when we look inside those vesicles, they're full of this decondensed chromatin. So if long ago you guys took molecular microbiology and you learned about DNA and they talked about the beads on the string of like the histones in the DNA, that's what this is. So you can see like the same thing you would see in these old textbooks. And so the neutrophil is packaging the DNA in these vesicles, and then it's releasing it in this process called like exocytosis, where it gets released without poking a hole in the cell. And then this is just showing what this looks like. So these are two neutrophils. So the blue is the nucleus of the neutrophil. The green is the Mycobacterium tuberculosis, because we've engineered the bacteria to express green fluorescent protein so we can see it. And then these yellow, and the red is just actin, so it's just showing you where the cell is. And in the yellow is that histone citrullination marker. And so you can see these vesicles that have this decondensed chromatin in them, and then they're being released as these webs. And so what we were able to do then is use genetics to determine what genes were essential for this process. And what we found was that this gene PAD4, or this gene PADI4 that encodes a protein PADI4, is absolutely required to get this histone citrullination and this net release. So if you delete PADI4, you infect with TB, you no longer see this response. So now we have a genetic way to block this net response. The other thing that we discovered was that there's another signaling molecule, type 1 interferon, which is really critical for controlling viral infections. But it turns out that in the context of TB, the type 1 interferon will actually affect the ability of the cells to release those webs. So we don't see any webs coming out in the absence of type 1 interferon. And I'll just say this is because that type 1 interferon, we don't see those vesicles of DNA. Okay, so then kind of jumping ahead one thing. So now that we had these molecular details, we could actually test whether or not blocking this process of net release would lead to better control of TB. And so we first looked at this in vitro in cell culture, where we took the neutrophils from a mouse that was either just a normal wild type mouse or deleted for this PADI4 gene, so it cannot make those nets, infect with TB, and look at the growth of the bacteria. And like we saw before, the bacteria grows great in wild type neutrophils. Mycobacterium tuberculosis loves neutrophils, but if you stop the ability of the neutrophil to be able to release these nets, we're able to inhibit its growth. So just by inhibiting that one process in the neutrophil, we can better control the replication of Mycobacterium tuberculosis. And now we want to see, is this druggable? And so we went into a mouse model of TB infection, and we inhibited that PAD enzyme using this compound, chloramidine. So we infect the mice through the aerosol route, just like a human would be infected. So they breathe in the bacteria, it goes into the lungs, the bacteria replicates in the lungs, and then we tried to treat the mice. And we first just looked to make sure that we were actually blocking what we wanted to block. So we were blocking that histone citrullination. And we did this by looking at lung sections, either vehicle treated. You can see that they citronate a lot of histones, release a lot of nets in response to TB. But if we use chloramidine to block that PAD enzyme, then we lose that signal. So we are effectively blocking the ability of the neutrophils to release nets. So does this impact TB pathogenesis? Yes. And so if we then look in the lungs at the TB burden, just by inhibiting this process for 9 days of our infected mice, we're able to significantly lower the bacterial burden, showing that chemical inhibition of net release can promote better control of TB. And so we think that one thing now that we're testing in conjunction with the TB Alliance for Drug Discovery, which is the Gates Foundation TB drug discovery arm, is whether or not we can take more advanced compounds that are able to block this process to better treat tuberculosis through a host-directed therapy strategy. And so that's just a little overview of what my lab is doing to tackle this problem. This is just showing the pathway and just showing that this response of neutrophils to TB leads to increased bacterial replication, tissue damage, inflammation, and granuloma and necrosis. And so we think blocking this could be a potential new therapy. And that's it. With that, I can take any questions. With population health? Yeah, so I think the work in my lab or mycobacteria in general. Okay, so For mycobacteria in general, I mean, I think that, again, so for the non-tuberculosis mycobacteria, I think there's a lot of opportunities because we have nothing in place. We have no public health, no implementation science, nothing going on with the non-tuberculosis mycobacteria, because we just haven't actually put together the infrastructure yet to actually monitor, again, like where people are getting infected. Are our interventions working? Like, are people adhering to it? How do we get more patients, you know, in a position that they feel comfortable coming to the clinic and taking drugs and things like that for these infections? So I think for the NTMs locally, there's a lot of opportunity because there's literally nothing that's been done, and we need to start thinking about that. For TB, I think of the public health initiatives as being a lot of public awareness, first of all, because in general, a lot of people in the US think TB has been cured. And so if you talk to anyone, a lot of people I tell that I work on tuberculosis, they're like, oh, why? But we already cured that. Or a lot of people think they've been vaccinated for it too, which they haven't if they've lived in the US their whole life. And so I think there's also just like a lack of public awareness as well in terms of the infection. And part of that is also disbarring some of the stigma as well, right? So like this is not an old or infection of the poor, like this is just an infection that infects a third of the world, right? And so like really getting that information out so that we can better monitor and treat those infections would be some ways.
So that was great. Thanks. Relevant to that, so much of what we do is through partnerships and collaborations with the community. And so I just wanted to know if you have any, I know you said you haven't, there's not a lot, and you want to have public health and population health and public awareness. Do you have natural partnerships, perhaps that you, or community advisory boards or groups that you're already working with that could be built on? Or are you looking for all of, starting from scratch? You were talking about starting from scratch. Are you looking from starting from scratch, or do you already have some of this in place?
So we would like to leverage things that are in place, but part of the reason that I'm here, and this actually would have applied to the last answer too, like I have some ideas, but I am by far not the expert, right? Like you guys are the expert here. And actually I was telling Sandra, I've been interacting with Ginger McKay, and just we've had like one or two conversations, and each time it's like this whole perspective and thought process that I hadn't thought of before, right? And so we don't have anything in place, but we think that we can leverage things that are already in place. But I also don't know personally what those are, right? And so I think that's the key to developing these collaborations, this network, me being here, is trying to figure that out, right? So getting people like you in the room to be like, okay, when you tell me this is the scenario, here's where I see the gaps are, and here's things that I think we should be working on.
I know about TB from a long time ago, but this was really, talk about learning, this was really impressive. I also don't know why anybody would follow that regimen. Seriously,
I mean, they have to here. Actually, if you don't take, depending on the city you're in, so I did like all my training in New York City, where they have like fair number of cases of tuberculosis, but an amazing public health system for TB. But they actually can quarantine you if you aren't taking your drugs. So there's like public health laws in place in some of these cities that force people to take them. But sometimes it's not the patient that, it's not the patient's fault, right? Like if they're getting really sick, maybe they just can't tolerate the antibiotics in some cases, and it's a real problem.
Yeah, I think a lot of times people adhere to what they can.
Yeah.
And it doesn't always match what we see.
Exactly. And I think another thing I didn't mention that we do in the US here too is we do prophylactic isoniazid treatment. And so if somebody tests positive for TB in the US, but they aren't sick, we generally prescribe them isoniazid, which is one of those frontline antibiotics, or now isoniazid and rifapentine for 9 months, for example. And that in itself is liver toxic. You can't eat red meat, you can't drink, some people don't tolerate it, and you don't even feel sick. You haven't even gotten tuberculosis. So you're just like, when I teach the med students, it's a very clinical lecture. And I'm like, you're gonna be at a point, you have someone who's completely not sick, feels fine, and you're gonna tell them to take an antibiotic for 9 months and makes them feel sick to prevent getting sick, hopefully. And so yeah, so these sorts of things are really hard in terms of patients, figuring out what's best for the patient. And really what we need is better drugs and better prevention methods, because it also isn't, it sucks that we have to have our patients do this too. Like you said, I wouldn't want to do that personally. Yeah. Oh.
Thank you so much. At the beginning of your lecture, you were talking about the drug supply issue being a barrier to successfully treating patients with these very long regimens. So as you're thinking about targets for future drugs, how have you started to think about how you'll overcome some of those drug supply barriers and access barriers?
Yeah, no, this is key for TB drug development, because you cannot make an expensive or difficult to mass produce drug. There's just no way it's going to happen. And so this is something that happens really further down the line. And luckily, there's a lot of acknowledgement of this from like the Gates Foundation and WHO. And so if you can get to a point that you have something that seems like in the optimization and the production stages of the drug can be minimized to like the cheapest possible and easiest possible drug development, then they will help you do that. But it is a really, really important point. And I think actually aligned with that point is the fact that then industry partners don't want any part of this. Because right, you're not going to make a money-making drug. You're making something to save lives. And so luckily there are some pharmaceutical companies that have put some effort into this. But actually one of the most recently FDA-approved drugs, the Daclin, came from Janssen, and they just pulled out of the TB space. And the Daclin has been great, although there are a lot of issues we can talk about. That no one screened for resistance before they rolled it out, and it turns out there's all this underlying resistance. And so it's also these sorts of things. But yeah, so there have been companies in there, but it's also getting industry partners incentivized to be able to do this when you're taking like their resources away from making another GLP-1, for example, right? And so I think like this is a challenge in terms of what you were asking about, like what do you do to get your drug cheap and be able to distribute it widely? But then you need partners for that. So what do you do to incentivize those partners? Because certainly my lab could not do it. No one could do it. Like you need a big industry partner to do it. So yeah, this is a big thing in the field. Thank you. Hi, Zoom.